for the computer. All right. So chapter one is the scope of microbiology. Okay. And one of the things that we need to understand, all right, is that microbiology was one of these things that didn't exist until the invention of the microscope, which was something that Leeuwenhoek came up with in the late 1800s as a way of looking at fabrics. But he was curious and he decided to look at living material. And what he found with every piece of living material that he studied, the first being a slice of cork, was that all living material was made of cells. Okay. And that was a major scientific breakthrough back in the late 1800s, right? And formed one of the two bulwarks of what's called cell theory, right? Which are what? Number one, all living things are made of cells. Okay. And number two, all cells come from pre-existing cells. And that was an observation made through the microscope as well. Okay. So I know that today we don't think of that as, I mean, we think of that as just common knowledge, but back then that was huge. We had never realized that before because humans believe what they see. And until we could see something, believe in it. And the microscope allowed us to see this for the first time. Okay. Before the advent of the microscope, all right, we believed a lot of strange things about things like disease. Like, for instance, if you were a citizen of the Aztec Empire and you were sick, you went to the doctor who had only two implements at his disposal, a hammer and a chisel. And the theory then was that if you were sick, it was because there was a demon living inside your head. So he would sit you down and take the hammer and chisel to your skull and pop a hole in your skull to let the demon out. Many of his patients died immediately. But, hey, that's all they had back then. In the Middle Ages, we thought the cure for everything was bloodletting because we thought that the health of the human body was determined by the balance of phlegm and bile and blood. And if you had balanced those correctly in the body, your health was okay. So if you were sick, you went to the doctor, who back then was also the barber and the dentist and the surgeon. So that should tell you something about the quality of care back then. And he would cure you by bloodletting. So he would cut you, or he would apply leeches and remove a certain amount of blood. And then after a certain amount of blood had been removed, he would pronounce you cured, and he would drive the bloody rags outside of his office on a white pole. And believe it or not, that is the origin of the barbershop pole. I know that seems a little strange, but there you go. Okay. And it wasn't until the microscope that we discovered a whole world that we didn't know existed before. And it led us to what we're going to discuss a little bit later, which are Koch's postulates, which is this idea that if you can isolate a microbe from diseased tissue, grow it in the lab, infect healthy tissue, and recreate the conditions of the disease and re-isolate that microbe, that's probably what's making you sick. And again, that seems like common sense today. But before the advent of the microscope, we couldn't do that because we couldn't see what we were looking for. Okay. So microbiology, the word micro, is referring to something smaller than the naked eye can see. A variety of techniques are used here for visualization, identification, and the study of microbial function. And the science originated with the invention of the microscope. So thank you, Mr. Leeuwenhoek. Okay. You can see some relative size here. Microscopy allowed humans to magnify objects and microbes not detectable by the naked eye. Advances have led to improvement of microscopes, which became essential investigative tools for biology in general and for the study of cells, tissues, and microbes. On the left, you can see the relative size comparisons of bacteria, eukaryotic cells, and viruses. And on the right, you can see a human cheek cell with bacteria on its surface, which are stained and appear as dark purple spheres or rods. Look how tiny they are. Okay. Look how small they are. And then think that a virus is 1,000 times smaller than that. Okay. I mean, we're getting really tiny here. There's a red blood cell, right? And there's an E. coli, it's the bacteria that some of the bacteria that lives in your colon and helps break down your waste products. There's a rickettsia, a very tiny bacteria. There's a pox virus, the biggest of the viruses. And then we get down to these other viruses, okay? And look how small they are. Look how tiny. We're talking the nanometer range, 10 to the minus ninth. And this is one of the reasons, guys, that all that masking up with paper masks for COVID-19 didn't work because the holes in the masks were far too big and the virus easily goes right through it. Okay. So everybody who was wearing those paper masks, it was basically symbolic. It was to make you feel better. It didn't do anything to halt the spread of the virus unless you were wearing an N95, which has holes that are too small for viruses to get through. So there's a lesson right there from science. You're looking at the size. It would be like me trying to say that, I could hold water in a colander or in a coffee filter. It's not going to work. Okay. The molecules are too small. All right. Zacharias and Hans Janssen were eyeglass makers and produced the first compound microscope around 1590, but it wasn't until Leeuwenhoek that we really took off here again, 1632 to 1723. So again, uh, he it was I should say 17th century, not 18th. He was from Holland, father of microscopy, observed protozoans and bacteria, and he called them animalcules. Okay. His interest in science and his curiosity led him to some of the most important observations of biology. He was able to see life forms called animalcules. Throughout the years, he observed bacteria, protozoans, blood cells, microscopic nematodes, rotifers, and more. Hooke was an English scientist that improved the design and capability of the compound light microscope and observed insects, sponges, protozoans, and more, and he drew them in a publication called Micrographia. Okay, so this is the first time that we got an artist's rendition of what you could see through the microscope. Okay. Light microscopes use visible light and optical lenses. They can be either simple or compound. The ocular is what you look through. The objective is what magnifies the specimen, which is on the slide, which just sits on the stage, which is above the condenser and the iris diaphragm and the light source. The final magnification is always the product of the ocular and the objective magnification. As an example, okay, if you were using the lowest power objective, which is a 4x objective, and you were looking through an ocular, the magnification of the ocular is always 10 times, so it would be a 40 times bigger image than the actual object. Okay, so dissection in stereo microscopes, the low powers for observing whole objects. Bright field microscopes, the background is lighter than the specimen. Most specimens require fixing and staining for bright field microscopy because they don't normally have color to them. Most microbes are transparent with the exception of those that are photosynthetic, which are green. Okay. Dissection and stereo microscopes are designed for observing larger objects like insects, worms, plants, or objects that may have to be dissected for further observation. These microscopes provide three-dimensional images to determine surface structures and locations of a specimen. Bright field is commonly used to observe sectioned and stained tissues, organs, and microbes. And there is an example of a bright field microscope. And you can see the different parts: the oculars, the objective, the stage, the condenser, the iris diaphragm, the coarse and fine focus, and the illumination adjustment or rheostat. And on the back there, there's an on-off switch as well. Okay. So let's learn a little bit about the microscope.
Hi, I'm Joelle, and I'm going to show you a few helpful hints on how to use the compound microscope. These pointers will help you use the microscope successfully. Before we begin, there are a few things to take notice of on your microscope. First, you want to make sure that the condenser knob is turned all the way up. Be sure not to confuse this knob with the focus knobs. You want to make sure that your fine focus is in mid-range. You can do this by lining up the white line with the edge of the coarse focus. Next, you want to make sure that your oculars are even. You can do this by setting your right ocular at 64 and your left ocular at 64. Next, you're going to make sure that your blue filter is in place. Lastly, you want to make sure that the 4x objective is in place. Now we are ready to turn on the microscope and begin. Turn on your microscope and raise the rheostat all the way to 10. Now we are ready to put the slide on the stage. For this demonstration, we're going to be using the cross-section of a human artery. Make sure that your slide is not full of fingerprints. If it is, you're going to take a Kim wipe and clean it off. Pull the stage clip open. Place the slide on the stage so that it fits into the corner of the slide holder. Release the stage clip so that the slide is held securely. You can then move the slide using the XY controls. Next, we're going to move the slide so that the specimen is centered in the path of light coming through the microscope. Open the iris and move your XY controls so that the specimen is centered in the light beam. Close the iris again so that when you look through the oculars, you do not blind yourself. Before looking through the oculars, you want to raise your stage all the way up. You would do this by turning the coarse focus knob away from you. You'll notice that the stage stops moving, but the coarse focus knob continues to turn. You are now ready to view the specimen through the oculars. You need to fit the oculars to your face, and you can do this by moving them in and out. This is difficult and may take some practice, so you may want to start by setting them at the 64 mark. As you adjust the oculars, you may see some funny effects. If your eyes are too close or too far away, you may see some circles jumping around. Keeping your eyes approximately 3/4 of an inch away from the oculars works best. Just think of it as looking into the distance into a pair of binoculars. Now, slowly move the coarse focus towards you until the specimen pops into view. You will also use your fine focus to sharpen the image. Once you have done this, you can use your XY controls to move the specimen around. What you see here is the cross-section of the human artery. The empty center is where the blood would flow. The pink section is the smooth muscle, and the purple strands are elastin. These are the effects of using the coarse focus, fine focus, and XY controls. There is one important thing that you must do before going to the next objective. You must place a desired part of the specimen in the center of your field of view. We can now move to the 10x objective. In order to see the specimen clearly, you may need to increase the light by opening the iris and using the fine focus to get a sharper image. Our next step is to view the specimen under 40x. Once again, you may need to make adjustments with the light by opening the iris and using the fine focus to get a sharper image. Going to the highest objective, also known as oil immersion, takes a few more steps. You don't need to use oil immersion with every specimen that you view, but we will go through it together. To place a drop of oil on the slide, you're going to move the objectives so that the specimen is centered between the 100 and 40x objective. You can then place 1 drop of oil right over the specimen. You can now turn to our oil immersion lens. You may want to adjust the light and fine focus as done previously. After viewing the specimen, you're going to want to properly clean the lens and the slide. To clean the slide, you must lower your stage and remove the slide. Then you will clean it with a Kim wipe. To clean your objective, you're going to turn the objective so it's facing out and clean it with a piece of lens paper. You have now learned how to use the compound microscope properly and successfully. It's now time to shut down the microscope. But before doing so, there are a few things you need to do to ensure the success of the next user. First, you're going to make sure that your 4x objective is in place. You're going to close your iris. Then you want to turn down your rheostat all the way back down to 1, and then turn off your microscope. That's it. Good luck in lab.
Now, a dark field microscope is used to view unfixed, unstained specimens such as living organisms. The background is dark, the specimen is bright, and motility can be easily observed because the organisms are alive. Some bacteria are difficult to stain as well, including spirochetes, and can best be observed by dark field. Bacterial capsules are good candidates for observation with dark field as well. So you can see what a dark field image looks like there. Okay. Those are cheek cells, human cheek cells. Okay. Phase contrast microscope is used to view unfixed, transparent specimens, effective for observation of cytoplasmic streaming, motility, and the dynamic states of cell organelles. Phase contrast microscopy uses high contrast images of transparent specimens such as living plant and animal cells, microbes, and thin tissue slices. And that's what it looks like. Okay. That's a phase contrast peek at a cheek cell. Okay. Fluorescence microscope uses UV light. Visualization of the specimens have naturally fluorescent substances or they've been stained with dyes. It's used in diagnosis of infectious diseases. It's useful in microbial ecology and applied to identify specific antibodies in response to particular antigens. You might, for instance, be looking for a particular surface molecule. You can treat that particular specimen with an antibody that has a fluorescent dye attached to it, and then you can hit that with a fluorescent light, and it will glow, like this picture of Pseudomonas. Okay, ultraviolet lamp illuminates the specimens stained with fluorescent stain. Okay. A confocal microscope generates sharper images, allowing visualization of different planes of a specimen using a laser for illumination. The image can be displayed three-dimensionally with electronic staining. The confocal imaging system is a single point of light focused sequentially across a specimen, avoiding most of the unwanted scattered light that usually obscures an image when the entire specimen is lit at once. So let's look at how a confocal microscope works.
Hey everyone, quick biochemistry basic here. Let's talk about confocal microscopy. Confocal microscope was invented because of drawbacks of fluorescence microscope. So what are the drawbacks of fluorescence microscope? In fluorescence microscopy, it uses a high intensity UV light. As the samples are continuously exposed to high intensity UV light, it causes photobleaching. As all the fluorescence molecules of sample above and below the focal plane gives fluorescence, the image obtained is blurred. So to solve these problems, a confocal microscope was invented. A confocal microscope is nothing but a modified fluorescence microscope with two major modifications. Number one, it uses a laser light instead of mercury arc lamp as a source of light. And number two, images are taken with a digital camera with a pinhole. What's the function of pinhole? Pinhole allows light of only one focal plane to be focused on the digital camera. Light rays coming from above and below the focal plane are cancelled out. Now let's see the working of the confocal microscope. Number one, laser is focused in a small region of the specimen. The fluorescent dye in the specimen gives fluorescence, which is captured by the digital camera. Number two, using two rotating mirrors in X and Y direction, the laser is now focused in the next region of the specimen, and the fluorescence is captured by the digital camera. Number three, the laser scans the whole surface of specimen and images of each spot are captured. And finally, number four, a software combines all these images into a one sharp image. Because the laser scans along the surface of specimen, confocal microscope is also known as confocal laser scanning microscope. The confocal microscope is also used for generation of 3D images. Because the pinhole allows the light from only one focal plane, the images of different focal plane can be taken and compiled into a 3D image using a software.
What do the images look like? Well, they're very detailed. Okay. Electron microscopes are sophisticated 20th century instruments. TEM is an electron beam that goes through the specimen, needing special prep, sectioning, and staining, produces a two-dimensional image with good internal detail. The pictures are referred to as electron micrographs. Okay. So this is even greater detail than the confocal. All right. Let's take a look at the electron microscope and how it works. Give it a minute. Hold on. Back out and hit it. That's how this goes. Try that. Okay, let's do this. Copy that. Let's go over here. Let's go over here. There we go. Just take a minute. Let's take a peek.
Science Talk, Professor Dean explains. We just learned all about light microscopy, which was the standard until the middle of the 20th century and remains tremendously useful today. But as we began to learn more about the microscopic world, using light microscopes that could visualize individual cells, we started to become curious about structures that are smaller than a cell, some details of which were difficult to visualize and study under standard light microscopes. Fortunately, electron microscopy, developed in the 1930s and 40s, proved to be much more powerful and allowed scientists to view these smaller structures. Some electron microscopy techniques employ a resolution of 0.2 nanometers, which is more than 1,000 times better than light microscopy. With this high electron microscopes are able to magnify their specimen up to 1 million times their original size, allowing researchers to study proteins inside of cells and the molecular structure of viruses, all of which led to a better understanding of the microscopic world. Of course, we have to ask, how do they do this? Well, the answer is in the name. They use beams of electrons, which interact with the sample as the beam is transmitted through the specimen. As we recall from our study of chemistry, electrons are stable subatomic particles that carry a negative charge. Electron beams have a much shorter wavelength than the visible light that light microscopes use, which we refer to as the de Broglie wavelength, as we learned in the Modern Physics series. This shorter wavelength allows for a higher level of detail and resolution in imaging. One type of electron microscopy is called transmission electron microscopy, or TEM. For TEM, specimens are fixed on a support grid and either embedded in resin or stained with a material like uranyl acetate or compounds of heavy metals like osmium tetroxide. Certain structures within the specimen will take up more of the stain than other parts of the specimen, and those sections are called electron-dense regions. When electrons pass through electron-dense regions, they lose more energy compared to when they pass through sparser regions. This phenomenon is used by TEM to visualize the specimen at high levels of resolution and magnification. For the stained specimen to be viewed by TEM, it must be dehydrated, embedded in plastic resin, and cut into ultra-thin slices with a glass or diamond knife. The sections must be ultra-thin so that the electrons can pass through them. When the specimen is secured in the specimen holder, an electron beam is generated and shot out of the electron gun. The electron beam is accelerated down the column of the microscope after passing through the anode. The electron beam then passes through the specimen, once again losing energy as electrons pass through electron-dense regions. The electrons then pass through the objective lens, magnifying the image, before the electrons pass through the projector, or ocular lens, which magnifies the image further and projects it onto the fluorescent screen. This is where we start translating the electrons into an image. The fluorescent screen is coated with a chemical which appears as bright spots when electrons come into contact with it. The intensity of the bright spot depends on how much energy the electrons have when they hit the fluorescent screen. The camera under the fluorescent screen then captures the image made from the bright spots, which is displayed on a computer screen. Transmission electron microscopes are used to visualize structures that are too small to study with light microscopes, such as the cell organelles, proteins, cytoskeletal filaments, and the components of cell membranes. But even with the unmatched resolution and magnification abilities of TEM, it still does have its limitations. Some of these include the fact that you cannot study living specimens or image 3D structures, because the specimens must be dehydrated and cut into ultra-thin slices before imaging. The second type of electron microscopy is scanning electron microscopy, or SEM. With SEM, specimens are coated with vaporized gold or palladium ions. Because when electrons come into contact with this coating, they cause atoms on the surface of the specimen to emit electrons. These emitted electrons are called secondary electrons, and these are what SEMs use to visualize their specimen. After the coated specimen is secured on the stage of the SEM, the process starts out similarly to TEM in that an electron beam is shot out and accelerated towards the specimen. In the SEM, the electron beam then sweeps or scans the surface of the specimen, which causes it to discharge secondary electrons. There are two types of detectors in the sample chamber: the secondary electron detector and the backscatter electron detector. The backscatter electron detector picks up backscatter electrons that come from deeper regions of the sample that are a result of elastic interactions between the beam and the sample. The results from this detector are used to look at crystallography and the magnetic field of the sample. But for the purposes of microbiology, researchers focus on the detection of secondary electrons, which allow them to visualize the topography of the sample, or the distribution of parts and features on the surface of the sample. The detectors convert the electron signal into a light signal that is amplified by passing through a photomultiplier before the final image is projected on a computer screen. SEM imaging is primarily used to study the topography of cells and other similar structures. With this type of microscopy, researchers are able to investigate the depth of structures, which isn't possible with TEM, as SEM produces a 3D image, opposed to the flat image from TEM. Yet, just like TEM, SEM cannot visualize live specimens, and SEM has worse resolution than TEM. Both TEM and SEM are expensive and require a lot of training, so they are not likely to be used apart from those pursuing a microbiology career. We covered quite a lot in this tutorial, so let's briefly summarize. Electron microscopy has around 1,000 times better resolution than light microscopy, because electron microscopy uses electron beams instead of light, which have a shorter wavelength. This allows some electron microscopes to visualize specimens at 1,000,000 times their original size. Transmission electron microscopy, or TEM, records the differing energies that electrons possess after they pass through a specimen stained with heavy metals to visualize small, detailed structures, like proteins inside of cells, and molecules associated with viral particles. Scanning electron microscopy, or SEM, captures the secondary electrons that are emitted from the surface of samples coated in vaporized gold or palladium ions to visualize the topography of the specimen. And the main drawback, as we mentioned, is that both SEM and TEM cannot be used to visualize living specimens, and they are also quite expensive and complicated techniques to use. And with that, we know a bit about electron microscopy. Thanks for watching, guys. Subscribe to my channel for more tutorials. Support me on Patreon so I can keep making content. And as always, feel free to email me, ProfessorDaveExplains@gmail.com.
So a much more expensive system, but much more detail. Okay. This is, for instance, the only way that we could see something like a virus. All right. So until the invention of the electron microscope, we didn't have a way of directly visualizing viruses. We could see their effects, but we couldn't see the actual viral particle itself because it's so tiny. Okay. Scanning SEM. Okay. Again, no section, scans the surface, gives a three-dimensional image. Magnification ranges from 10 to 100,000 times larger than the object. You can see an example there, SEM image of some Campylobacter. Okay. Atomic force microscopes have an advantage over SEM in that the surface of interest doesn't need a metallic coating. It's a finely balanced stylus that touches the surface of the specimen and is moved over its surface, traveling up and down as it follows the contours. The contours are then compiled into a topographic image. Now, again, before we realized there was a microscopic world out there, we had some ideas about where life came from, and the two competing theories at the time of Leeuwenhoek were abiogenesis and biogenesis. Okay. In abiogenesis, they proposed that life developed from non-living matter on a routine basis. The proponents believed that living things can come from non-living material. The opponents started doubting and developed experiments to contradict this view. Pasteur ended the controversy with one of his famous experiments using the swan-necked flask. Okay. Francisco Redi, in support of spontaneous generation, maggots from fly eggs. Leeuwenhoek, in opposition. Animalcules gave fuel for each side of the debate. Needham, turbidity in boiled broth, claimed victory for abiogenesis. Okay. But it wasn't until Pasteur's experiment that we had some supporting evidence for biogenesis. Okay. Spallanzani repeated Needham's experiment without air access to the flask, and he found that there was still growth because there were some organisms that were sealed in there before. He cut off the air supply. So again, that was support for abiogenesis. But it was Pasteur and the swan-necked flask experiment that really gave us supporting evidence for biogenesis. Okay. He had two swan neck flasks. He took broth and superheated it inside the flask. Okay. Sterilizing everything in there. All right. And then on one flask, he broke the neck off, and the other, he left the neck intact. The flask with the intact neck did not show turbidity, no microbial growth, while the flask that had the neck broken off showed microbial growth. Okay. And so that was big supporting evidence for this idea of biogenesis. Now, to be fair to the abiogenesis people, they were right at least once, which is wherever the first cell came from. Okay. And that could have been on this planet, could have been somewhere else. We don't know. All right. But it does not happen on a routine basis on this planet. Okay. Living things come from pre-existing living things. The germ theory of disease was one of these things that we had ideas about before the invention of the microscope, but the microscope gave us conclusive evidence. The germ theory stated that suspicion that microbes cause not only spoilage and decay, but infectious disease. Oliver Wendell Holmes linked childbirth deaths to dirty hands of midwives and physicians. Semmelweis linked maternity infections to contamination of hands and required chlorine solution wash, and Lister expanded protocols with aerosol disinfection, introducing a septic technique. Okay. During the time when the existence of spontaneous generation was being debated, several doctors began to suspect that microbes not only could cause spoilage and decay, but might play a role in infection. Koch, investigation of anthrax, convinced the existence of infectious microbes was a real thing, and he developed Koch's postulates. Koch discovered that anthrax endospores persist after the death of the exposed animals. He proved that these spores could survive and later develop into active anthrax microbes and infect other animals. This established that a specific organism caused a specific disease. After years of experimentation, he developed Koch's postulates, which set forth conditions that should identify an organism as the specific cause of a specific disease. Jenner was the father of immunology, and he was able to demonstrate that you could vaccinate against a particular disease using extracts of the infectious agent, okay, by using cowpox to inoculate against smallpox. Jenner was able to demonstrate this conclusively, okay? Koch's postulates state that the microbe must be present in every animal with the disease and absent in healthy ones. The microbe can be isolated and grown in pure culture outside the host. The microbe must cause the same disease in the inoculated animal. The same microbe must then be isolated from the infected animal, okay? And those are the postulates. Now, there are some cases in which the postulates break down, all right? For instance, there are some microbes that cannot be cultured, all right, yet they can cause disease, okay? There are some microbes that can be that can only be grown under specific circumstances, such as in the organism in which it produces a disease, such as in a human, okay? And there's no way to do that ethically, all right? And there's some instances in which the microbe cannot be observed under a light microscope, all right? But these days we can detect its genetic material, okay? So there are places where Koch's postulates break down, all right? Now, one of the things that science likes to do is to categorize the world around us in order to understand it better and to make better use of the information that comes in as it's new, okay? The origin of microbes is described in geological time. The fossil record of prokaryotic life suggests that microbes were perhaps the first living thing on Earth. Prokaryotes go back up to 4 billion years ago, while eukaryotes go back 2.2 billion years. Now, what do we mean by prokaryote and eukaryote? Okay. A prokaryote does not have a membrane-bound nucleus and has a simpler cellular architecture, while a eukaryote does have a membrane-bound nucleus and has a much more complicated cellular architecture with membranous and non-membranous organelles, while prokaryotes only have non-membranous organelles, okay? So again, we get into terminology here, right? Karyo means nucleus, pro meaning before, eu means perfect. Okay. So prokaryote before the true nucleus, eukaryote with the true nucleus. The other type of prokaryote, besides bacteria, are the archaea. Okay. They don't have a membrane bound nucleus, but they turn out to be more closely related to eukaryotes, of which we are a member, than they are the bacteria. Okay. And we know that through the comparison of their genetic material. Okay. And then we have pathogens that are non-living, okay, such as viruses and toxins, prions, okay. So when we talk about microbes, it's better to look at them as you can have living microbes and you can have non-living microbes, okay. A non-living microbe flunks the conditions for life, which are what? The ability to grow, the ability to reproduce, the ability to respond to your environment, the ability to move, the ability to change over time, organization, and cellular structure. Okay. And with viruses, viroids, and prions, we can cause disease, and we're a microbe, but we are not living because we flunk two of the tests of life, which is that we can't reproduce on our own. Viruses need a host, as does a prion, right? And we are not cellular. Okay. So we're not living, but we can produce disease for sure. Okay. Now, taxonomy is just this idea that we can organize the world around us using similar traits. Okay. We've broken down the living world into the broadest category, which is domain, beneath that kingdom, phylum, then class, order, family, genus, species, and strain. Binomial nomenclature established by Linnaeus has a genus and species designation for every living thing on the planet that is specific for that living thing. Escherichia coli, for instance, for some of the bacteria that live in your colon. Escherichia is the genus name, coli is the species name. You always italicize it in print, okay? And if you're writing it in script, you underline it, okay? So the idea here, okay, is to make the information of the world around us more useful, okay? It's a little bit like your mail. When your mail comes, you can throw it all into one pile. And then when you have to find something, you have to sort through everything to find what it is you're looking for. Or you can, as the mail comes, split it into categories, such as bills, and then another pile for personal correspondence, and then another pile for junk mail, right? And then you can reach into, say, the bill pile, pull out anything in the bill pile, and you immediately know without even looking at it that it's something you need to open up and pay or the service will be discontinued. Okay. And so that's beneficial, right? Because that allows us to make better use of the things that are in those categories and understand them immediately because they're in those categories. Okay. So let's take a look at classification.
Taxonomy. It's the science of classifying living things. That sounds exciting. Today, we'll basically be learning the Dewey Decimal System of evolution. It's like filing. Must be on the edge of your seat. Okay, shut up. When it comes down to it, this science doesn't just categorize organisms. When you look a little deeper, you realize it's telling the story of all life on Earth, and it's a pretty good story. Every living thing on this planet is related to every other living thing. If you go far enough back, we all have a common ancestor, an organism that both you and I are descended from, or something that a starfish and a blue whale are both descended from, or even weirder, that an oak tree and a salmon are both descended from. That organism lived. It lived very long ago, but it was here. And I dig that. The trick of taxonomy is basically figuring out where all those branches of the evolutionary tree are and finding some convenient labels to help us understand all of these remarkable interrelationships. Let's be clear, though, taxonomy isn't about describing life in all of its ridiculous detail. It's mostly about helping humans understand it because it's way too complicated without structure. To get that structure, biologists use the taxonomic system to classify all the organisms on the Earth. It's sometimes called the phylogenetic phylogenetic tree, or the tree of life, and it illustrates the evolutionary relationships between all living species. So there are about 2 million known species, but there could be anywhere from 5 million to 100 million species. Scientists really have no freaking idea. New species keep getting discovered all the time, and the more organisms we have to keep track of, the more complex the phylogenetic tree becomes. So there's not always a consensus about how to classify this stuff. There's a lot of gray area in the natural world. Actually, let me rephrase that. The natural world is one giant gray area. Sometimes it's just hard to know where to put a certain group of organisms, and eventually the group gets so big the classification system has to be messed with to make room for it. So the system isn't perfect, but it's good enough that we've been using it for around 250 years. What's that? Do you smell a biography coming on? Carl Linnaeus was a Swede, born in 1707, and early in his career as a botanist, he realized that the botanical nomenclature of 18th century Europe was, well, just crap. For instance, in his day, the formal name of a tomato plant was Solanum caule inerme herbaceofolus pinnatus incisus racemus simplicibus. Linnaeus actually said, "Once I shudder at the sight of most botanical names given by modern authorities." Not only did the sloppiness bother him, he saw a whole sugar storm blowing in, because new plants were still being discovered in Europe, but that was nothing compared to the crazy stuff that was coming from the New World. Linnaeus saw that pretty soon naming conventions were just going to collapse under all these new things to name, and then what? So Linnaeus famously started off by naming himself. He came from a peasant family, and at that time surnames were just for rich people. So when Carl went to college, they asked him for his surname, and he just made one up, Linnaeus, after the linden trees that grew on his family's homestead. Linnaeus got a medical degree and became a professor at Uppsala University, where he devoted himself to the study of nomenclature. He had his students go to places and bring back specimens for him to study and categorize. The method he eventually adopted was based on morphology, or physical form and structure. This wasn't necessarily a new idea. Back then, people grouped organisms by analogous or homoplastic traits, structures that appear similar but actually come from completely independent origins. By this definition, birds would be more closely related to butterflies than to reptiles, because both birds and butterflies can fly. Linnaeus had a good mind for this stuff and turned out to have a real knack for choosing actual homologous traits for his classification system, traits that stem from a common evolutionary ancestor. Linnaeus, of course, didn't know jack about evolution. Darwin wouldn't come around for another 100+ years, but he intuited that some traits were more important than others. For instance, he was struck by the fact that reproductive apparatus seemed to be a good way of classifying plants. He also caused a bit of a scandal by classifying class Mammalia based on the female abilities to produce milk from their nipples, because apparently that was pretty racy stuff back then. In his lifetime, Linnaeus cataloged roughly 7,700 plants and 4,400 animals. He published his classifications in a catalog called Systema Naturae, which by the time he wrote its 12th edition was 2,300 pages long. In the meantime, Linnaeus actually adopted a personal motto: God created, Linnaeus organized. Although taxonomy has come a long way since Linnaeus, we still use a bunch of the conventions that he invented. For instance, we still arrange things into taxa, or groups of organisms, and we still use the same taxa as Linnaeus: kingdom, phylum, class, order, family, genus, and species. We also still use Linnaeus's convention of binomial nomenclature, using a unique two-part name for every species, the genus and its species name, in Latin or sort of Latin-ish. This practice actually started back in the Middle Ages, when educated people were expected to know Latin. We know a lot less Latin now, but we know a lot more about evolution, which Linnaeus didn't. And we have technologies like genetic testing to classify relationships between organisms, and yet we still use Linnaeus's morphology-based system because genetic evidence generally agrees with classifications that are made based on structure and form. However, because there was a lot of life that Linnaeus had no idea about, we had to stick a new taxa above Linnaeus's kingdom. We call it domain, and it's as broad as you can get. The domains are bacteria, archaea, and eukarya. The bacteria and the archaea are prokaryotes, meaning that their genetic material goes commando with no nucleus to enclose it, while the eukarya make up all of the life forms with a nucleus and include pretty much all of the life that you think of as life, and quite a lot of the life that you don't think about at all. It might seem like since all macroscopic life only gets one domain, it's kind of silly to give prokaryotes two. And for a long time we didn't divide them up into different domains. They hung out together in a single domain called Monera. But it later became clear that bacteria, which live pretty much everywhere on Earth, including inside of you and deep in the Earth's crust, and archaea, which are even more hardy than bacteria, have distinct evolutionary histories. Archaea being more closely related to eukaryotes, and yes, thus me and you. They have totally different cell membranes, and the enzymes that they use to make RNA, their RNA polymerase, is much more like ours. Under the domain eukarya, which is by far the most interesting and even occasionally adorable domain, we have kingdoms Protista, fungi, plantae, and animalia. Now, scientists have settled on these four for now, but these are categories that are a human creation, but there are good reasons for that human creation. The unscientific truth is that we looked at life and divided it up based on what we saw. So we were like, well, protists are single-celled organisms, so they're very different from the rest of the domain. And plants get their energy from the sun, and fungi look and act very different from plants and animals. And, we already know what animals are, so they have to get their own kingdom. And those scientists are sometimes loath to admit it. That system of just looking and dividing things up actually worked pretty well for us. Not perfectly, but pretty well. But there's a reason why this worked so well. Evolutionarily, there are actual categories. Each of these kingdoms is a huge branch in the tree of life. At each branch, an evolutionary change occurred that was so massively helpful that it spawned a vast diversity of descendants. Plants, or plantae, are the autotrophs of the domain eukarya. Autotrophs meaning that they can feed themselves through photosynthesis, of course. They're cellulose-based cell walls and chloroplasts, giving them a distinct difference from all other multicellular life. There are two other sorts of trophs. There's the heterotrophs, which get their energy by eating other organisms, and the chemotrophs, which are weird and crazy and only show up in bacteria and archaea, and they get their energy from chemicals. Now, the kingdom Protista is weird because it contains both autotrophs and heterotrophs. Some protists can photosynthesize while others eat living things. Protists are basically a bunch of weird eukaryotic single-celled organisms that may or may not be evolutionarily related to each other. Scientists are still trying to figure it out. Some are plant-like, like algae, some are more animal-like, like amoebas, and some are fungus-like slime molds. Protists are one of those gray areas I was telling you about, so don't be surprised if by the time you're teaching this to your biology students, there are more than 4 kingdoms in eukarya. Fungi, which are, the funguses. They include mushrooms and smuts and puffballs and truffles and molds and yeasts. And they're pretty cool because they have cell walls like plants, but instead of being made of cellulose, they're made of another carbohydrate called chitin, which is also what the beak of a giant squid is made of, or the exoskeleton of a beetle. Because fungi are heterotrophs, like animals, they have these sort of digestive enzymes that break down their food and get reabsorbed. But they can't move, so they don't require a stomach for digestion. They just grow on top of whatever it is they're digesting and digest it right where it is. Just super convenient. And finally, we have Kingdom Animalia, which is the lovely kingdom that we find ourselves and 100% of adorable organisms in. Animals are multicellular, always. We are heterotrophic, so we spend a lot of time hunting down food because we can't make it ourselves. And almost all of us can move, at least during some stage of our life cycle. And most of us develop either 2 or 3 germ layers during embryonic development. Wait for it. Unless you're a sponge. So like I said, we use this taxonomic system to describe the common ancestry and evolutionary history of an organism. Looking at the phylogenetic tree, you can tell that humans are more closely related to mice than we are to fish, and more closely related to fish than we are to fruit flies. So how about we pick an organism and we follow it all the way through the taxa, from kingdom to species, just to see how it works? I know. Let's pick this kitty, because I know she'd like it. Right, cat? So kitties have cells that have nuclei and membrane-surrounded organelles, and they're multicellular and heterotrophic and have 3 germ layers of cells when they're embryos, so they're in the kingdom Animalia. And they have a spinal cord running down their backs protected by vertebra and discs in between them, and they have a tail that doesn't have a butthole at the end of it like a worm, which I'm really glad about. And that puts her in the phylum Chordata. Kitty clearly does not like this, so I'm going to put her down now. And the kitty lactates. It gives birth to young like a cow instead of laying eggs like a chicken. And they have fur and 3 special tiny bones in their ears that only mammals have. So they're in the class Mammalia. So she's more closely related to cow than to chicken. Good to know. And like a bunch of other placental mammals that eat meat, like weasels, the mustelids, and dogs, the canines, kitties are in the order Carnivora. And they're in the cat family, Felidae, whose members have lithe bodies and roundish heads, and except for cheetahs, retractable claws. And they're littler than tigers and panthers, which puts them in the genus Felis. And then at the level of the species, the descriptions get pretty dang detailed. So let's just say that you know what a cat is. So the species name is Catus. And look at that. Felis catus.
Oh, gee, I could have that whole thing cross-stitched onto a pillow for you to sleep on. You would be cute.
Thank you for watching our taxonomy episode of Crash Course Biology. We hope that you learned something. Thanks to everybody who helped put this episode together. If you have any questions for us, please leave them on Facebook or Twitter in the comments below, and we will get to them hopefully very quickly. We'll see you next time.
The Woolsey-Fox system generated 3 domains based on largely molecular evidence: bacteria, archaea, and eukarya, which include the protists, the fungi, the plants, and the animals. Okay. We've organized the classifications into ever more specific groups, starting with domain, then kingdom, phylum or division, class, order, family, genus, and species. The way to remember this is to remember dumb King Philip came over for good sandwiches. You can see here the breakdown of the kingdoms of life. The domains are the large colored areas, okay? And then the kingdoms within it, all right? And then the phyla and so on, okay? So you can see here, for instance, with bacteria, right? The domain is bacteria, phylum, proteobacteria, class, alpha, beta, gamma, delta, epsilon protobacteria, right? And then within the gamma protobacteria, let's see here. Yeah, gamma. You've got chromatiles, trocales, legionelles, pseudomonales, vibrio Enterobacteriaceae, and pastorellase, okay? And then if we follow the Enterobacteriaceae down, Enterobacteriaceae, genus, we got Enterobacter, Escherichia, Klebsiella, Proteus, Salmonella, Serratia, Shigella, Yersinia. Within the Shigella, you've got Shigella boydii, Shigella dysenteriae, Shigella flexinare, and Shigella soniae, right? So from broadest to narrowest, right down there with species, all right? And the benefit of doing this, of course, is that if you are in a particular group, all right, then you have all these characteristics assigned to you automatically, and then anything new can be added on, okay? The taxonomic resource for bacteria is Berge's Manual of Systemic Bacteriology, the second edition of which is published in 5 volumes. In addition to the number of keys for bacterial ID, newer versions of the manual contain molecular sequencing information for different groups, okay? Microbial ecology is the study of interrelationships between microbes and their environment. Microbes are everywhere in the environment and generally have an impact on maintaining ecosystems. Ecological interactions between organisms in a community, including mutualism, commensalism, synergism, and parasitism, are classed according to the degree of benefit or harm they pose to each of the participants. In mutualism, both organisms benefit. In commensalism, the waste product of a microbe provides useful nutrients for another organism. In synergism, 2 organisms are dependent on each other to break down a nutrient that neither can break down on their own. And in parasitism, one organism benefits while the other is harmed. The normal flora provide protection against pathogens by preventing attachment to the host tissue and competing for the same nutrients. If the normal flora is successful, the pathogens can't flourish or colonize and therefore are unable to reach sufficient numbers to cause disease. Foodborne disease result from consuming food that's contaminated with different pathogens of bacteria, virus, parasite, or microbial toxin. Waterborne disease is a general term describing diseases acquired from contaminated water supplies, resulting in 4/5 of all illnesses in developing countries and a high infant death rate. Airborne diseases are transmitted from infected people by coughing, sneezing, or talking. Pathogens are in small mucus saliva particles suspended as aerosols. Applied microbiology is the human use of microbes to improve certain aspects of life. Microbes are used in this capacity by the food, pharmaceutical, and agricultural industries, And in forensics and other endeavors, application of new technology and genetic engineering has increased the industrial use of microbes. A lot of non-pathogenic microbes occur naturally in food, such as in cheese and yogurt. Wine is produced by yeast fermentation of carbs in freshly harvested grapes, peaches, berries, pears, and other fruits or plants. In beer production, barley and other grains are used as the source of fermentable carbohydrate. The microbial content of drinking water should be constantly monitored to make sure the water is free of infectious agents. Pasteur observed the inhibition of microbes by products formed by other microbes. This phenomenon is called anabiosis. The discovery of the penicillin-producing mold penicillium by Fleming in 1920 started the successful search for other antibiotic-producing microbes. Okay, so one of the things that we have to appreciate here, okay, is that microbes are everywhere. They're all around us. They're in us, on us, and around us all the time. The bulk of them are beneficial or at least not harmful. And it's just a small percentage that are actually pathogenic. And that's what we're going to focus on this course. Okay. Um, Bioremediation is the process of using microbes to clean up toxic, hazardous, or unmanageable compounds by breaking them into harmless compounds. Some microbes performing these tasks have been genetically engineered to clean up waste or pollutants. By a process referred to as bioconversion, microbes can convert biomass, such as organic matter and human, agricultural, and industrial waste, into fuel, including ethanol, methane, and hydrogen. Microbial forensics is a relatively new field applied to solving bioterrorism cases, medical negligence, and outbreaks of foodborne disease. Okay, so the applications here are, I mean, they're everywhere. Okay, what you guys are going to be concerned with primarily is medical microbiology. In other words, how do microbes contribute to disease? What is the disease process like? What are the characteristics of these microbes? What are the signs and symptoms of the diseases that they produce? And how do we control them in a clinical environment? Okay. All right.